The next speaker is Dr. Brato Chakrabarti from ICTS. He will be talking about active carpet model for intracellular flows.
Hello, so I'm Brato. I recently joined ICTS. I worked at the interface of fluid dynamics, soft matter, and biophysics. And I will tell you a problem which involves all these three things. This is work done primarily in collaboration with Olenka Jain, Stas Schwartzman, and Mike Shelley. So humans have roughly 10 trillion cells and 200 different cell types. Yet, if you look closely, that there is a very little variation in the cell sizes, with majority of the cells being in the range of 2 to 30 micron. Now, what is quite interesting is that this cell size variation is true in a mouse and also in elephants. So this suggests that there is some sort of evolutionary pressure on being the right size. And this can be understood by realizing that for cells to function, you need to transport or move things around. And for that, it relies on the inherently slow process of molecular diffusion. And perhaps this sets a constraint on how large a cell can grow. But there are exceptions. X cells or oocytes are one of the largest known cell types in the animal kingdom. They can be as large as 500 microns to millimeters. And clearly, you need a different mechanism of transport in these large cells. One way nature solves this problem is through cytoplasmic streaming. So streaming is not new. It was discovered around 250 years back by the Italian scientist Bonaventura Corti when he was looking at these algal plant cells called Cara corallina. And in the cells, which can be centimeters long, he observed a persistent circulatory motion of the fluid all day, every day. And this persistent motion is known as streaming flows, which is rather generic in many other large cells. So I will tell you about one such streaming flow observed in the egg cells of the fruit fly Drosophila. So the egg cells or the oocytes of this fruit fly develops over a span of 80 hours. And towards the later stages of its development, which you see over here, it becomes a gigantic cell of size 500 to 600 microns, that's shown here. And you see an emergence of a vertical flow that spans the entire cell chamber. So this is the streaming flow that I'm thinking of. You can probe it experimentally. You can perform PIV to quantify its structure. Now you can ask that how these flows are emerging. So we believe that this is a result of fluid structure interaction phenomena. So the boundary of the egg cells are coated by thin hair-like micron-sized filaments called microtubules. These are anchored on the boundary, and there are molecular machines or motor proteins called kinesins that move from the base of the filament to the tip, carrying cellular payloads or cargoes. So as they move up, they push down on the filament. So these filaments are under compression. If the load is sufficiently large, they can buckle. And as these filaments buckle, they generate more flows. But now there are thousands of such filaments, so they all interact synergistically through the flows and the deformations. And this results in a self-sustained collective dynamics where you see the emergence of this large-scale flow in the interior, and all the filaments have laid down at the boundary like seaweeds. So what I want to tell you is about a coarse-grained model that sort of tries to put this description in maths and tells you about the organization of the flow in 3D. So in this model, which I'm calling the active carpet model, I'm not thinking about dynamics of individual filaments. Instead, I'm going to characterize a microtubule bed by a surface density C0 that tells me about our filaments per unit area, and a surface polarity field N that tells me about the orientation of these filaments at any particular position. To make my life simpler, I will think of these filaments as rods that are anchored to the cell surface, equipped with a torsional spring at their anchoring point. So this is really mimicking the bending response of clamped filament. If you perturb them, these springs will bring them back to its unperturbed configurations. And the actions of these motor proteins is coarse-grained into an uniform force density along the backbone. So what is the structure of the problem? This is like a boundary layer problem. We have a thin layer of active stresses coming from this layer of microtubules. These stresses drive flows in the bulk, but then these flows can alter the orientations of these vectors or the microtubules, and that again feeds back to the stresses. So they're all coupled with each other. You can write down an evolution for the polarity field. The first piece is called a Jeffreys equation. It tells you how a rod sort of orients in presence of a flow, which is U, which I need to compute. And then there's another piece coming from this torque, or the torsional spring, which wants you to be pointing radially inward. So to compute this velocity field U, I think of the cytoplasm as an incompressible Stokesian fluid. There is a no-slip boundary condition on the cell surface. And the action of this entire microtubule bed has been coarse-grained into a surface traction jump F bar across this interface delta S formed by the tips of the microtubule vectors. This stress has two pieces. First is active, which is very simple to understand. As the motors move, they push along the polarity direction. How much do they push? That depends on the density and the forces from the individual guys. And then there's another piece which is more subtle that accounts for the orientation and inextensibility of this whole fiber bed. So let me tell you what is the primary mechanism of what's going on. If filaments are pointing vertically upwards, this is a half-space problem I'm thinking of, then there is no flow in this mean field limit. But if you perturb it, then you generate a tangential shear stress at the interface, and the shear stress drives a shear flow in the boundary layer. So the shear flow will like to rotate these filaments, but now there is a competition between the rotation and the ability of the springs, which wants it to be vertical. And if the stresses are sufficiently large, you can get deflected in one direction. There are really two parameters, the density and the activity, which is just telling you how much forces you're putting in the problem. If you vary either one of them, then you transition from a state where you have no flow through a pitchfork bifurcation to states where you have flows, which I'm calling streaming states. So you can simulate this whole problem. I'm doing this simulation on a spherical egg approximation. So I'm color-coded the surface of the sphere by an order parameter that is 0 when the filaments are pointing radially inward and close to 1 when they're aligned. You start from this state, you immediately form regions of high alignment. You see the emergence of topological defects. You let it go, then these aligned regions start interacting with each other. This defect merge. And at the steady state, you're left with 2 + 1 defects sitting on 2 opposite poles of the sphere. You can look at the interior streamlines. The -1 defects are associated with hyperbolic points in the flows, and the +1 defects are centers. And the steady state, you have a system-sized vortex emerging inside the cell. So we can be a bit more quantitative about this whole flow field. The flow field is strongly vertical, but there is a secondary swirl component that you see over there that comes from the filaments pointing radially inward near the defects through which they channel fluid. In the plane of the vortex, which is the plane transverse to this defect axis and passing through the equator, the flow is entirely planar. And if you look carefully, you find that this velocity increases linearly, almost linearly from the center, which is signature of like a rigid body rotation. But now you have a no-slip boundary condition, so there is a shear flow in this boundary layer. And now you can do this PIV measurements from experiments, and you see very similar profile, and there is not only a qualitative, but fairly good quantitative agreement between a simple model and its measurements. So with this model, you can do much more analysis. You can sort of work out the stability results inside the sphere analytically. You can do weakly nonlinear theory and try to understand why the dynamics is low dimensional. You can do sort of prove what are the global attractors of the problem and so on. I won't go into the details of that. But if you're interested in this, there's a preprint available on arXiv on this work. And just to sort of tell you about some future direction, just like cows are not spheres, so are not eggs. In the late stages, eggs are actually elongated, like prolate spheroids that you see over here. And geometry clearly has an important role to play in organization of these sort of flows, which is what we're exploring with Olenka. So that's all, and thank you.
Nice talk. Just as a curiosity-driven question, what kind of flow you are expected to have if you do this kind of calculations for a cylindrical tube? This is inspired by the physiological problem that inside our respiratory tract we have cilia which propel the mucus.
So okay, the cilia is a bit different from this problem, right? In this problem, what is happening is that there is cilia is internally actuated. This microtubule is not. So here, really, the instability is taking place because of this buckling of the whole bed. But we have done this problem inside the cylinder, actually. And inside the cylinder, of course, you just get a swirling flow in the interior. So you have a rigid body rotation plus a shear flow. It looks, so whatever the flow you'll see in the plane of the vortex, this is the flow that you get in the cylinder. And you can compute the steady state analytically in that case.
Thank you.
Any further questions?
So you don't find cells in which the local arrangement of the cilia has defects in it, and so that.
So there are cases where local arrangements of cilia has defects. So for example, in the starfish larva that you see, so they are coated with cilia. They have very dense ciliary bands, and there are points where you find -1 defects in sort of ciliary arrangement. The part that I'm a bit unsure whether those defects emerge spontaneously from synchronization or whether there is more neuronal control to it, because in many of these cases, there is neuronal control to the ciliary carpet, and maybe it could be the control of the carpet which tries to generate different flows and the different organization. That's why you have defects. So it's not, I'm not sure what's the mechanism of that. But in sort of, if you have cilia on a surface, then you can imagine that just through synchronization, if you define a synchronization order parameter on the surface, you will have defects on that order parameter.
Right.
So that, which is different from the orientation bit. Yes.
Let us thank the speaker.